That's when we're celebrating our vision research. And I would like to welcome all of the soldiers of vision research here today. I know there is Arvo and a lot of people still traveling today, but I'm pretty sure those who join will enjoy a fantastic scientist and great research conducted in his lab. With that, I will pass to Arun for introduction.
Good morning and welcome to the Distinguished Speaker Series. My name is Arun Wu. I'm a project scientist working with Dr. Paul Chesky. It's my honor to introduce our guest speaker, Dr. Jonathan Lin, an ophthalmic pathologist and professor at Stanford University. Dr. Lin received his bachelor's degree in biochemistry at Harvard College in 1992. Then in 1999 and 2001, respectively, he graduated from Columbia University with a PhD in neurology and an MD degree. After completing his residency training in pathology at Brigham Women's Hospital in 2003, Dr. Lin completed his fellowship in Howard Hughes Medical Institute Eye Pathology at the University of California, San Francisco in 2006. He served as a clinical instructor in the Department of Pathology at UCSF until 2007. Dr. Lin has been serving as a staff physician at VA San Diego since 2008. In the same year, Dr. Lin started off as an assistant professor before becoming a full professor in the pathology department at UC San Diego in 2017 until 2021. Since 2019, Dr. Lin has served as a professor in the Department of Pathology and Ophthalmology at Stanford University, also serving as a staff physician at VA Palo Alto. Dr. Lin's research is focusing on the understanding of how misfolded protein-induced ER stress causes human diseases such as vision loss and neurodegenerative diseases. These studies led to the discovery of novel disease variants in the ER stress regulatory genes. His lab utilizes iPS-derived neuronal and retinal organelles, knock-in, knock-out, and transgenic mouse models, and antisense nucleotides and AAV gene therapies. He has published prestigious journals such as Nature Genetics, Human Molecular Genetics, and PNAS. Dr. Lin's remarkable achievements have earned him numerous awards. Just to name a few, he received the Excellence in Science Young Faculty Award from the American Society for Investigative Pathology in 2009, New Investigator Award from the Hope for Vision Foundation in 2009, Vision Research Award from Karl Kirchgessler Foundation in 2010, and Outstanding Investigator Award from the American Society for Investigative Pathology in 2021. Today, he will be giving a talk titled Targeting Protein Quality Control to Treat Eye Diseases. We are honored to have him speak with us today. Without further ado, please welcome Dr. Jonathan Lin.
Great. Can you see the screen?
Yes.
Great. So many thanks to Chris and Gratznia for the kind invitation to speak at your seminar series. I'm actually still at Arvo in Seattle, so I'm not from my desk, just on a laptop. So just let me know if there's any funny hotel internet connection issues. But what I'll do in this seminar is tell you about a mechanism, protein quality control mechanism, that through the course of this talk will illustrate that I think it's a gene agnostic, cell agnostic, tissue agnostic mechanism that can be involved in the pathogenesis of many different kinds of eye diseases and has potential to modulate treatment of these eye diseases. So the scope of my talk is I'll just briefly go over protein quality control mechanisms, and in particular, focusing on the best understood signal transduction pathways that control protein quality, the unfolded protein response, integrated stress response. Then I'll touch upon three different eye diseases where we believe these protein quality control mechanisms are important: retinitis pigmentosa, achromatopsia, as well as neovascularization, and just illustrate how we think these are important in the pathogenesis, as well as treatment opportunities arising from modulation of these pathways. So the mechanism that I'll tell you about related to protein quality control is based on the fundamental mechanisms regulating protein folding in the endoplasmic reticulum. This is an organelle present in all eukaryotic cells, metazoan cells, that is a protein folding factory for membrane and secreted proteins. It's a double membrane organelle, and it extends throughout the cell body in epithelial cell types, and it's devoted, a main function of it is devoted to ensuring membrane and secreted proteins are properly folded, and then when they're properly assembled, exported to their destination, either extracellularly or into the membrane to function properly. Because the quality of the fidelity of protein folding is so critical for their function, it stands to reason that nature's evolved protein quality control mechanisms to make sure that everything is functioning properly with respect to the protein folding organelles. And one mechanism that's well studied involved in this in the endoplasmic reticulum is the unfolded protein response. These comprise intracellular signal transduction pathways that, with sensors embedded in the ER membrane, shown here as cartoons, IRA1, PERK, and ATF6, they have luminal domains that monitor protein folding processes in the endoplasmic reticulum. And in response to anything that perturbs the function of the ER, whether it's a misfolded protein, nutrient deprivation, oxidative problems that interfere with disulfide bond formation, so on and so forth, these sensors can detect defects in the endoplasmic reticulum functions really, really well by processes that still are not very well understood. And in return, they signal across the membrane to initiate intracellular signal transduction cascades, just shown here by these little cartoons. And the net effect is that these signal transduction cascades activate transcriptional programs within the nucleus. They activate a lot of genes that help the ER fold proteins, help the cell and the organism survive protein folding stress, ER stress more effectively. They also activate translational proteins to begin to regulate the synthesis and biogenesis of new proteins, where in general, most translation is suppressed, but then there are some stress-regulated mRNAs who bypass this translational suppression and instead are upregulated when this pathway is activated. So this is a fundamental protein quality control mechanism that reprograms transcriptional programs as well as translation to help cells survive ER stress, protein folding, as well as the entire tissue and organism. A question that I've been interested in the eye realm is how this signal transduction system is clearly within all cell types. We all know from vision research, going to Arvo, stuff like that, that the cells in the eye are extremely metabolically active. Some of them secrete really, really intricate, massive amounts of proteins. How do these pathways that regulate protein quality control, how do they contribute to function and vitality of retinal cells? And then in respect to eye diseases, how do they participate in those processes as well? So I'll just talk a little bit about a couple different diseases where we think this process is quite important. The first disease is inherited retinal degeneration, achromatopsia. This is just eye pathology picture from the macular region, and the cones are at the bottom. They're extremely important, as everyone in this audience knows, for vision in people, and achromatopsia is a disease where the cones are dysfunctional, and it's a genetic disease, autosomal recessive, and the children have defects in cone-mediated vision as early as can be detected. And because it's an autosomal recessive disease, it facilitates the use of genetics, either classical genetics or sequencing genetics and kindreds to identify genes. So working with, in collaboration with the two achromatopsia group, Susanna Colin, Bern Bissinger, 10 years ago, approximately 10 years ago, we identified mutations in protein quality control that were found in families with achromatopsia. And this is just one of the protein quality control mechanism cartoon on the left, and various mutations in different kindreds on the right. And just to remind you then, what is this ATF6 protein that was identified as a achromatopsia disease gene? Well, it's one of those unfolded protein quality control stress regulators that sits at the ER membrane, shown here in blue. And in response to ER stress, it's activated to create a transcription factor. This is just showing ATF6 alone in more detail, where it normally is in the ER membrane. And in response to stress, it migrates to the Golgi, where it's cleaved to release a transcription factor that upregulates stress-induced genes whose products are thought to help cells overcome the stress. So that was what's known about the mechanism in all cell types. And what was really intriguing is why it's causing this congenital inherited retinal degeneration. So simple things to do was, since we had access to the patients and they were very happy to participate in studies with the medical retina specialists who are seeing these patients, we were able to directly study cells, generate retinal organoids from these families. This is just a kindred studied by Stephen Sang and Elise Hyun, where we asked fundamental issues. Well, how are these mutations in these families impacting this signal transduction mechanism? And you can see the mother, who's number 1 on the top right panel, and the 3 children who have achromatopsia. You can see the protein, the achromatopsia defect of ATF6, it doesn't function very well as a transcription factor. It's about 50% the level of the wild type, what's seen in the parent. Looking at it in more detail in just purely recombinant proteins, so independent of any other related transcription factors that could be interfering with it, we again showed with recombinant disease-related achromatopsia proteins that transcriptional activity was completely abrogated. This is just a limited targeted sequencing panel where the wild type transcription factor, you see robust induction of a dozen different genes, 20 different genes, but 3 disease variants were just quite inert, unable to, unable to function as a transcription factor, but also not as a dominant negative, anything like that. So they were just ineffective as a transcription factor. So we had some biochemical insight into how this was affecting the protein. And the next issue was, how is this leading to achromatopsia, loss of function? To do that, we took advantage of, again, the patients who had provided all their fibroblasts, and we wanted to model see if we could study this a retina in a dish using the retinal organoid technology. And we were able to successfully recapitulate the retinal organoid technology, develop it, and we were able to identify a really obvious defect, which is really quite pleasing. Just to remind you that the retinal organoids are kind of like a retina, but with the outer retina on the surface of the retinal organoid. And then what's really nice is that photoreceptors, they have the outer segment structures that protrude out. So you can really study photoreceptors quite readily. And something that was really interesting evident to us in our diseased retinal organoids from these patients was that the retinal organoids, they differentiated, stem cells differentiated fine and everything, but just by light microscopy examining the development of the retinal organoids, there was a surface morphologic defect where all the bumpy protrusions that were present on the outer surface, this is just a wild-type retinal organoid at 9 months, where you see these bumpy protrusions that are labeled with cone opsins in green. All these bumpy protrusions were missing from our diseased retinal organoid. This is just a normal retinal organoid on the top, and then the diseased retinal organoid. So they were same size and volume, but there was a loss of all these bumpy protrusions, which at this time point is almost primarily cone outer segments. So this suggested to us that we were able to model a defect that could be leading to the achromatopsia phenotype shown in the patients. This is just some molecular confirmation staining of these section retinal organoids, where the wild type, you see the cone opsins, the peanut agglutinin markers, and then in one of our patient samples, the cone opsins are gone and the agglutinin is much flatter and reduced. Gene sequencing also confirmed that there was a defect in the cones. These are just cone markers, which were completely downregulated in mutants compared to wild type. And so putting this all together, we were able to successfully recapitulate cone defects in this retinal organoid model. And this really provided a mechanism for a platform for us to begin to investigate pathomechanisms. How does ATF6 regulate cone development, especially with respect to the outer segment, inner segment cone-like morphology, but also to begin to test therapeutics to rescue this defect that was found in our retinal organoids. Before moving to that, we also went back to the patient's retinas in collaboration with Joe Carroll to see if they also had this similar defect in morphology that was seen in our retinal organoids. Specifically, Joe did adaptive optics on the patients carrying this mutation, and you can see at the level of screening with his adaptive optics, they're really focusing on the outer segment, inner segment, the cone-like morphology. And you can see in the normal patients, they really have a cone-like structure. And all our achromats, the structure, the mosaic was gone. That was suggestive of being replaced by rods. So kind of similar phenotype where the morphology was completely abrogated in our retinal organoids, and AO suggested that that was also the case as well in the people. And then using this platform, then we begin to see if we can test therapeutics. One approach that we thought about was using the conventional, the really popular gene editing technology. Since this is an inherited retinal degeneration with, a DNA mutation, all the gene editing technology would be one approach and many people are doing that to correct these gene mutations. These, as you know, they're by gene editing, base editing, CRISPR editing, so on and so forth, then delivered into by viral vectors into the cones or rods of interest. The Luxturna has been extremely successful, but it's really expensive, and it's really gene dependent. You really have to have the gene editing tailored just for that specific mutation. So this is going to be a really affordable approach. So we wanted to explore other possibilities. And we thought, well, this is a fundamental ATF6 and protein quality control. It's a fundamental pathway. The signal transduction pathway is well worked out, and it's involved in protein quality control. Why don't we develop a drug-like approach to target this pathway and restore its activity? And so in collaboration with Luke Weissman and Jeff Kelly at Scripps Institute, they did a screen of 600,000 compounds for molecules that could restore function activate this pathway artificially in a drug-like manner. This was just done in a luciferase screen. They had to do a lot of counter screens, transcriptional profiling, and they end up with really simple cell diffusible, cell permeable, small molecule compounds shown here on the right-hand side, which is really inexpensive and could be used for experimental purposes. And this molecule activates a signaling transduction pathway by this mechanism shown here. where normally the inactivated ATF6, it's in the endoplasmic reticulum, and then in response to stress, then it migrates out of the ER to form the transcription factor. What the drug does is it promotes the migration, the exit of this protein out of the ER so that you end up with more transcription factors. And this was a really attractive mechanism of action for this molecule to treat our diseases, because from looking at the biochemistry and cell biology of these mutations, what we noticed was that a number of them, they were all defective with respect to their transcriptional activity, but the way they caused a defect, a large number of them caused a defect because they kept the precursor form of the protein, the full-length protein within the ER. They were kind of stuck within the ER, unable to migrate out and form the transcription factor. So we have patient mutations that lead to loss of function by keeping the precursor protein form in the ER. And we have a drug that promotes exit from the ER. So we thought this would be a really great match with respect to mechanism to use these drugs to push out the disease variants. And we just tried something really simple. Oh, this is just in biochemical studies. We're looking at recombinant proteins, the wild type ATF6. You can see the transcriptional activator form, the cleave form, formed really robustly. But with the DTT, the artificial stimulant, it doesn't form very well at all. But with the drug form AA147, it also forms the cleaved transcription factor form really robustly. But in the disease form, then you see the appearance of the transcriptionally activated form, which was not responsive under normal stress conditions. So we had our retinal organoid model, and we had this disease model, and we had this small molecule that could target the pathomechanism involved in this disease. And we just did something really simple during COVID. We just added this molecule to growing retinal organoids to see what would happen, whether it would cause toxicity, whether it would cause rescue, whether it would turn the retinal organoid into like pancreas or something crazy. And we were really pleasantly surprised that it's non-toxic in our systems, and others have shown that it's non-toxic in mouse models. I showed it restored the biochemical activity. And in our retinal organoids, we begin to see what we think is therapeutic rescue effects. First, we saw increase in cone-related gene expression, and then looking at the morphology, the surface of the retinal organoid, it is really evident that we begin to see bumpy protrusions again, compatible with regrowth of the photoreceptor structure. So this was really, really promising to us, and we're still studying this to understand how it's regulating structure formation of the outer segment, and also optimizing this molecule to find the best time points to treat the retinal organoids with intent that this could be a way to help photoreceptors form their structure. And with respect to these patients carrying these achromatopsia ATF6 mutations, it would be a really, really targeted genetic therapy that's quite affordable, that's could be orally available and bioactive to help their photoreceptors grow back. And so in summary, in this little vignette, what I've illustrated to you is one disease where we think protein quality control is causal for this eye disease. Specifically, we have a mutation in a protein quality control gene that leads to achromatopsia. I've suggested a couple of different therapies. There's the gene editing therapy to correct IRDs. And then what we're really interested in is more of a gene agnostic therapy using pharmacologic manipulation of proteostasis to treat this particular disease. The next disease scenario I'll touch upon, it's another IRD, the retinitis pigmentosa. And this is targeting the rod system of the outer retina. And as this audience well knows, one of the most common causes for retinitis pigmentosa are defects in the rhodopsin protein. This is a classic G protein coupled receptor. Chris has done amazing work elucidating its structure and function. And it's super, super important for photo detection, transduction. And as everyone knows, hundreds of mutations in this little small G protein coupled receptor lead to retinitis pigmentosa, slow decay of the rods, and there aren't any treatments for it. So it's one of the more common IRDs, and it's still lacking treatments. Rods produce a huge amount of it. The most common mutation in North America is the P23H mutation. Many groups have shown that this mutation, along with many other missense mutations in rhodopsin, lead to protein aggregation, folding of rhodopsin, just shown here in a dose response on the left, wild type glycosylated rhodopsin versus P23H rhodopsin. It's all aggregated, dimerized, oligomerized. And this was a really attractive molecule to interact with the protein quality control systems, especially the ER protein quality control systems, because the biochemistry of it being misfolded and located in ER was so good. And then also there's the ER protein quality control pathways. So we and many other people took advantage of first asked, well, is this activating the system? And we took advantage of fluorescent GFP mice, crossed it with these, and it was really, really clear that this was activating it quite potently, where you can see in the wild type mice, the rods, there's very little GFP expression, whereas in the mutant mice, a lot of GFP. And this has become a really fun tool. A lot of people are using it. Marina Gorbachev, Stephanie Hagstrom, really, really fun tool to rapidly detect at the cellular resolution what cells are experiencing ER stress within the retina, especially in mouse models. So we have really good evidence that this is a misfolded protein, and it's triggering especially this protein quality control pathway. And then the natural function as well, what is that? How is this signal transduction mechanism? How is it participating in the pathogenesis of the disease? And can it possibly be co-opted to help these disease rods? So many people, including our group, did very simple approaches using Chris's mouse, where we begin to manipulate it, cross it with all the mouse mutants in these protein quality control genes. And this is work of a lot of it work from people like Michael Cheatham, Sarah Zhang, Marina, myself, and others. And many different genes, protein quality control genes, have been crossed with the P23H mice or with the P23H or other P23H model organisms. And the punchline is deleting at least these main sensors it's detrimental. It accelerates P23H retinal degeneration, rod photoreceptor cell loss, and function. And so this is telling us that the protein quality control pathways are really important for rods to survive this sort of protein folding and inducing stress from P23H rhodopsin. And so what therapeutic opportunities could there be then, knowing that these are involved in the metabolism, especially the catabolism, probably degradation of the mRNA as well as the protein? Well, the clear opportunity is then, since loss of these is accelerating retinal degeneration, perhaps activation of them could be beneficial to restore protein homeostasis in the rods. And there's work along those lines from Marina early on, where she overexpressed one of the downstream targets in, I think, the P23H rat model, and that showed some beneficial effects. And I think what the way going forward with protein quality control for rhodopsin mutations is that this is a relatively slow progressive chronic disease. Rods are really, really good at eliminating degrading these mutant proteins, mutant rhodopsins. This is just shown here in the middle panel, where the steady state level of P23 rhodopsin in homozygous mice is very, very little compared to wild type. So they're really, really good at it, especially in contrast to epithelial cells, which normally don't see rhodopsins, and you can see steady state levels really, really well. But this is a progressive disease where you can have where the degradation is occurring in the ER regulated by these UPR pathways. And there can be events throughout life that can lead to a secondary stresses, whether nutrition deprivation, environmental conditions, coexisting diseases. And in that situation, what probably happens is that the UPR system, ER stress becomes too much because you already have the endoplasmic reticulum and these protein quality control pathways are already working really, really well in the heterozygous people to get rid of rhodopsin mutant rhodopsins really well. If you have any events that increase stress on top of that, then that could overwhelm those rods and lead to the kind of this progressive dropout you see over time. So with that in mind, I think what one wants to do is kind of boost these pathways, these protein quality control pathways, so that during bouts of stress and periods, it can still cope with P23 rhodopsin as well as the new sources of stress and not accelerate, and then allow the photoreceptor expressing P23 rhodopsin to still keep degrading P23 rhodopsin, but also survive that secondary sort of stress at that period. So kind of like a chronic, kind of like a vitamin sort of format to improve protein quality control, I think would be really beneficial for this particular disease. The last clinical disease vignette I'll talk about is how these protein quality control pathways can participate in another set of eye diseases related to pathologic retinal neovascularization. And this is different than all the photoreceptor diseases I talked about. And it relies on the fact that the human retinal anatomy is super well organized. The retina is fed by two vascular sources, the outer retina where all the photoreceptors live. It's fed by these choriocapillaris in the choroid, shown here on the very bottom, these little blood vessels. The inner retina, where the retinal ganglion cell lives, it's fed by a separate blood supply in people, the interretinal vasculature. And you can see with the precise organization of the human retina, that now everything has to be precisely in place. In pathologic retinal vascular diseases, and what happens is the blood vessels break through, they're disorganized, they grow in the wrong spots, they bleed, they leak, and in doing so, then this is really detrimental to all the neurons in the eye, by leading to scarring, by disrupting their access to nutrition and blood supply, and it underlies many vision diseases. So pathologic retinal neovascularization and diabetic retinopathy and wet AMD, retinopathy of prematurity, some of the common diseases, and also many inherited retinal neovascularization diseases like MACTEL, macular telangiectasias. So there was a lot of evidence that I mentioned to you this protein quality control pathways, they initiate transcriptional and translational programs to reshape the cell to survive stresses. There was a lot of intriguing evidence pointing to these stressways being involved in angiogenesis, inducing angiogenesis as a response to stress. And that makes a lot of sense because increasing blood supply and nutrition will help cells fold protein, in a very general sense, help cells fold proteins better, survive stresses better. So it makes sense that a lot of angiogenesis genes are stress related. And we explored this possibility in particular with the ATF6 mice, and also others have done it with other transcriptional factors like XBP in this system, because in other systems, it was shown that the important angiogenesis-related genes like VEGF, HIF1, were transcriptionally or translationally regulated by these pathways. So how could we explore whether these pathways were involved in angiogenesis in the eye? We took advantage of the knockout mouse, in particular the ATF6 knockout mouse, which we are studying for roles in photoreceptor diseases. It's viable, it doesn't have any obvious phenotypes. At baseline conditions, the eye looks has good vision, the vasculature looks almost entirely different. The other genes are harder to study, are and harder to study in that the knockout mice are lethal or peri-lethal, so they're harder to study because one would have to make conditional specific deletions to study retinal vascular sort of problems. So we started with the ATF6 mice and just examined how it looks normal under baseline conditions, but would there be any changes under conditions that cause abnormal blood vessels to grow? And we took advantage of the oxygen-induced retinopathy model. This is widely used to create abnormal leaky blood vessels. And what one does is just put the developing mouse at a time when the retinal vasculature is developing, change the oxygen levels so that the blood vessels grow too fast or too little. And in doing so, by growing abnormally, they start to leak, as shown here by neovascularization, form scars, and lead to vision loss by damaging the retina from all that leaky blood vessel abnormal scar formation. And this can be quantified and looked at. These are just retinal flat mounts from the wild-type mice where you see the amount of obliterated blood vessels at different times under these procedures. Changing oxygen levels can suppress blood vessels, vaso-obliteration, and you can get neovascularization later on as a compensation for all those leaky blood vessels. But then there's always going to be all these scars, so the vision is going to be all deranged because of that. And we looked at what happened to the ATF6 mice under those same conditions. And by quantifying the amount of vaso-obliteration, neovascularization, we were able to see a striking reduction in the growth of pathologic blood vessels in the absence of this proteostasis factor ATF6, just shown here quantified on the right-hand side. The neovascularization is much less by histology. The function also supported that the neovascularization was less because the vision scotopic and photopic recordings were much improved in the ATF6 mice after this challenge. The blue recordings are from the wild-type mice where there's a lot of damage after in this OIR pathologic neovascularization models, and the red traces are from the mutant mice where you can see the retina is much more responsive, more alive and functional after this challenge. The sequencing confirmed that a lot of this was due to defects, changes in angiogenesis processes in the absence of ATF6. These are all the terms, some significantly enriched terms, and a lot of them were related to blood vessel formation and endothelial function. Looking specifically at these enriched terms, if they were induced or suppressed in the mice, this is just a violin plot with some of these endothelial cell terms, and the violin plot shows that there's a significant median reduction in the median expression of endothelial terms. And some of the specific genes by qPCR show suppression, including like the VEGF marker. So this is showing that without this proteostasis compound, transcriptionally and probably translationally, a lot of endothelial genes and terms are not functioning properly. Last, with the histology, looking at the retinal vasculature, this is the inner vasculature because most of the OIR is damaging the inner retina vasculature in these mice. Looking at the number of proliferating endothelial cells in wild type, just looking at the triple labeled, there's a number easy to detect. And in the mutant mice, there's significantly fewer proliferating endothelial cells as shown by these markers. So in this vignette, then we show that with this proteostasis compound, it's present in all cells, and it's important for cellular survival to ER stress, protein folding, quality control. With degenerative diseases, it's playing a positive role in maintaining the health and viability of photoreceptors, keeping them alive and functional. But at the same time, these protein quality control pathways, they can also be detrimental. They can promote the survival of cells that are bad, that you wouldn't want to have be around. For example, in this case, the proliferative endothelial cells that create all this disruption and pathologic neovascularization diseases. So in summary, in this talk, what I've illustrated then is gone over protein quality control mechanisms, focusing especially on the endoplasmic reticulum protein quality control pathways. talk a little bit about 3 different disease processes and diseases where we think that they're playing an important role. 2 of them are inherited retinal degenerations. One of them is a broad pathomechanism involved in pathologic neovascularization in many different retinal diseases. And then there's work from many, many other groups and other eye diseases implicating these this same pathway as well. The glaucoma field work, including from Gulab at UCI now, as well as Yang Hu, Jeff Goldberg, has implicated these protein quality control pathways in experimental forms of glaucoma, especially the optic nerve crush injury, where the damaged retinal ganglion cell axons release huge amounts of calcium and trigger massive ER stress. And in particular with the glaucoma field, therapeutic and chemical and genetic ways to block these ER stress caused by all these damaged optic nerves seems to be really, really powerful in experimental forms of glaucoma. Also in other parts of the eye, like in the anterior cornea diseases, there's extensive work showing that ER stress, UPR signaling could be involved in the pathogenesis of Fuchs endothelial corneal dystrophy. And in summary, then, what I hope to show you with this talk is that we believe protein quality control and these mechanisms, it's involved in the pathogenesis of many different eye diseases, both positively and negatively. And it can be thought of as a really interesting, attractive therapy to treat these diseases in that it's gene agnostic, it's cell agnostic, and it's tissue agnostic. And you can think about activating these pathways to treat diseases like the photoreceptor diseases to improve their viability, development, and survival. At the same time, inhibiting these pathways can be useful for other kinds of eye diseases when you have cells that are not supposed to be growing, like in retinal vascularization diseases. And by blocking these pathways, then that could be a way to suppress the growth of those diseases and treat different kinds of eye diseases. So these are the folks that in my group who participated in the projects. And for all the different eye diseases, we've had the pleasure of working with many, many different collaborators for all the different achromatopsia, retinitis pigmentosa, as well as the chemical gene studies with long-term collaborators, medicinal chemists at Strips. And with that, then I'll take any questions, and thank you guys for your attention.
Jonathan, they couldn't be better explain what you have shown today. So thank you very much for that. Can you hear us?
Yeah.
So I have 2 questions, and it's perhaps you lost me somewhere. The ATF6 mutation, you have about 12 or so mutations that were on one of your slides. And the compound which enable export of the ATF fragment out of fragment of whole thing out of ER to Golgi, they are not very close to the cleavage site, right? They are just spread out throughout. So, and you mentioned that you wanted to have agnostic, not specific to a particular mutation compound. So is your compound extracting entire protein out of ER, the transmembrane part as well?
It's not extracting the entire protein, but what it is doing is it's pushing the entire protein out of the endoplasmic reticulum. And by doing so, it goes to the Golgi, where there's a Golgi-specific protease that cuts that full-length protein in half and releases the transcriptional factor to go into the nucleus to function. And so, yeah, it's more kind of just pushing the protein out. And we like that mechanism as a treatment for some classes of these mutations, because some classes of the mutations, they keep the protein, make it sticky and keep it stuck in the ER so it doesn't migrate out very well. Yeah.
So this would means that again, what we have is export of the full protein, and then the cleavage happens not in ER, but in Golgi.
That's right. So the drug itself is more kind of pushing the protein out of the ER with respect to the disease, some of the classes of disease mutations, it's attractive because they get stuck in the ER. And then we also think it's going to be fun for other disease mutations in photoreceptors, like some of the CNG channels or whatnot, to push some of the subunits further down secretion just to restore this. So you have a little bit more protein, subunit receptor proteins, whatever at the surface that might restore some function as well, just by enhancing the secretion of proteins. It could be beneficial for other sorts of things that get stuck in the ER.
I understand. Hopefully, I understand it. So this wild type ATF6 is also cleaved in Golgi. Is that correct?
Yep, that's right.
There's no cleavage at all in ER.
That's correct.
Okay, and my second question before we go to web, and we have a few people who wanted to ask question there. The second question I have, so if you have your organoids, do they experience also a stress, the folding pathway stress?
Yeah, there's our sequencing supports that there's in the absence of
Any mutation.
This in the absence of protein quality control genes, they're going to experience increased stress. And what's really fascinating to us is that it kind of the stress is manifesting as, I think, a morphologic it's a morphologic problem during development of the photoreceptor outer segment, inner segment sort of structures, where we think the cell is formed, like the recoverin labeling is present, and the nucleus and the immediate soma is present. But then the ability to form the structure, the inner segment, outer segment, cone-like, even rod-like structure is a very, very challenging task, requiring secretion of membranes and proteins. And during that period, these protein quality control systems are super important. And we think that without ATF6, and that's why cones are susceptible. Yeah.
All right, let's go to Vladimir and then Cheryl.
Hey, Jonathan, thank you so much. Very
Hi.
interesting work. And also thank you for joining us while traveling for ARVO.
Yeah,
A couple of questions. So the ATF6, if I followed your presentation, it seems to be cone-specific or to be targeting predominantly cones. Can you comment on the expression of ATF6 in rods and why it seems to affect
absolutely.
cones?
Yeah, so it's actually a ubiquitous protein. It's present in all cells, and but the defect, so the mutations are present in germline in all cells in the human body. But the defects in the people carrying homozygous disease alleles or compound heterozygous disease alleles manifested first as achromatopsia. So the cones are super, super sensitive to loss of this protein. It's emerging that there's other cells in the body that are also sensitive to loss of these proteins, although at maybe slightly different periods. So we have work showing that there's families with cone rod dystrophy, so where the cones die first, but then the rods are sick later as well. ATF6 can be mutated in those families as well. And then more recently, in work that's in revision right now, we've shown that hair cells in the ear cochlea are also really, really sensitive to loss of ATF6. They will decay as well, leading to a hearing loss defect in some of these ATF6 patients with achromatopsia. So what's common between all these cells that seem to be vulnerable? Well, cones are clearly the most vulnerable, but also rods and hair cells in the ear, is that they all have really, really polarized, specialized morphologies. And it kind of suggests to us that ATF6 is just really important for the proteins or lipids or structure of these specialized cells. Yeah.
But rods are not as affected. So do you have any idea why cones and not so much rods?
Yeah, I speculate a lot about, and also the other thing is, mouse pods, ATF6 mouse cones don't have this defect as well. They're perfectly functional at early times. So not only is it that cones are vulnerable, to the or require this protein, but it's human cones in particular. And so we have a lot of speculations. One thing that idea that I really am intrigued with is the calyceal process. Or the cone has a really, really bizarre outer segment sort of structure, and there's some evidence that the calyceal process in cones human cones is really important, and that's kind of like this membrane proteinaceous actin secreted sort of crown-like thing that supports the outer segment. There's some evidence that the human cones have they're bigger cones compared to mice, and perhaps the calyceal process, ATF6 is really important for the formation of this calyceal process to support development of the human cone outer segment structure. Other ideas that we have is with the interphotoreceptor membrane compounds, composition and secreted things that may be important for providing, which is made out of these secreted proteins that may be important for the development of cone outer segments. And so without ATF6, they're not forming very well. Yeah.
Okay. And my second question is there's emerging evidence that extracellular vesicles are another way by which photoreceptors seem to handle misfolded proteins. So do you have a sense of the relative role of the unfolded protein response versus EVs in maybe handling
P23? Yeah, that's super fun. Yeah, so the unfolded protein response, it's really targeting, regulating things at the endoplasmic reticulum level. And so I think of the EVs maybe a little downstream of that, where the vesicles bud off from the endoplasmic reticulum, and most of the time they should go to the Golgi and then also go to the outer segment to form the rhodopsin disc membranes. But maybe sometimes the extracellular vesicles that are full of, extracellular vesicle, the EVs that are full of P23H, it would make it kind of be attractive that the photoreceptor would know that this P23H rhodopsin, you don't want to put it into the outer segment discs. It's defective to get secreted and get rid of it. I don't know. I speculate that these pathways would promote more activation and that would promote more secretion. But then what it's unclear to me is how would the cell photoreceptor know specifically to target a vesicle, distinguish a vesicle full of wild-type rhodopsin versus a vesicle of P23H rhodopsin, or in people, it's going to be, compound, some P23H and some wild-type. How would it know which vesicle to secrete versus correctly incorporate into the connecting cilium and put into the membrane? So I think that there's some really fascinating basic biology questions of rhodopsin trafficking and recognition of P23H rhodopsin, identification of P23H rhodopsin versus wild-type rhodopsin. And I think that this ER will definitely play a role in the secretion and generation of these vesicles, but with respect to the specificity and selectivity of identifying P23H and versus wild type, I think that's a really important question, and that really should be studied. Yeah.
Okay, thank you.
Yeah.
Sure.
Good morning, and it was an excellent talk. I enjoyed it a lot. One of the questions, can you hear me?
Mm-hmm.
Okay. One of the questions that you were talking about, gene therapy versus the small molecules and targeting with a small molecule, those people that have the ATF6 knockout in patients. And I wondered if you had other small molecules, if you'd have to repeat the injection, and how effective it might be long-term for solving the
absolutely. Yeah, so we tried it with we've performed our experiments, published experiments with one small molecule that modulates protein quality control. We have, there's a lot of other ones that, I showed one of those drug screens where there was like 600,000 compounds, and we ended up with one, but there's actually obviously a lot of other ones that look attractive as well. And so we're testing some of these other compounds for rescue in these organoid sort of models right now. Then with respect to ultimately, how would we apply this to people? So, then intraocular injection is one approach. obviously, but we hope that these small molecules could be taken as a pill, orally bioavailable once a day pill. The reason for that is there's other diseases that would used a small molecule proteostasis modulator approach that were successfully translated. These are diseases that amyloid sort of diseases where they develop small molecule modulators to stabilize and clear amyloid, misfolded amyloid proteins, and are FDA approved tafamidis. And they formulated it, ultimately they formulated as a once a day orally bioavailable pill. And so that's kind of what we would hope long-term our research will lead to, that these small molecules, which are self-permeable, bioavailable, could be developed into an oral pill. And I like that idea too, because the ER system, it's working throughout life. So what we really want to do is just kind of enhance and boost its function under durations of distress, periods of distress, to keep it well functioning, kind of like in a vitamin sort of or supplement sort of format. And by doing so, especially with these chronic progressive diseases, I think it will could have a positive impact. Yeah.
Hello, Dr. Lin. Thank you so much for your wonderful talk. I have a question about the P23H associated RP. It was really fascinating to see that you can reduce the ER stress, in your study. But then, I have a question about the total levels of rhodopsin on the disc membrane, because even though you were able to reduce the P23 misfolded, P23H proteins in the ER, still there are reduced amount of rhodopsin on the disc. And then the rhodopsins are also structurally important for maintaining the disc membrane and structure. So do you have any thoughts about it?
Yeah, so I think you guys can educate me about this. It's not clear to me that the discs with P23H rhodopsin are as viable as wild type discs. It seems to me that the cell rod really wants to, it really wants to get rid of P23H rhodopsin, and is devoting a lot of energy, especially like with these UPR pathways. It seems like the role of all the knockout studies are supporting that it's positive, it's getting rid of P23H rhodopsin either at the transcriptional, translational, or degradation post-translational level. Putting more P23H rhodopsin left, it's not clear to me that will be the best way to treat it. With respect to disease where it's a heterozygous condition, I think making sure there's no P23 rhodopsin would be a sensible approach, in that patients that are just heterozygous for rhodopsin, so they have one mutant allele, one null allele of rhodopsin and just one viable rhodopsin, they're fine and stuff like that. But it seems to me then that making sure there's no P23H rhodopsin and P23H retinitis pigmentosa would be a sensible approach, because then if it works really well, it would kind of be like people with just one copy of regular rhodopsin who are fine. Yeah.
Hi, Dr. Lin.
Does that make any sense? Or
Yeah.
Hi, Dr. Lin. Thanks for a clear talk. It's Philip Kaiser. So I was looking at the AA147 molecule, and my understanding from the experiments that you've done, those are all sort of in cell culture. Have you, this is following on Dr. Kraft's question, have you looked at the oral bioavailability of this molecule in mice and kind of its half-life?
Yeah, so our colleagues have done A147 studies in mouse with tail vein injections, and shown efficacy in, I think it was brain and heart disease sort of models, especially with the brain models being able to cross the blood-brain barrier. So So it seems like it's effective in these mouse IV sort of injection studies. More recently, what we've done is just intravitreal injections with with all of these compounds actually, and we're just and we have evidence looking at safety and efficacy parameters for all these different compounds, and we've kind of evidence prioritized list of the most effective and potent sort of compounds. But in those sorts of studies, we have evidence as well that A147 is safe in the eye and can modulate transcription as well. So yeah.
Thank you.
Great.
No, one more question.
Hi, Jonathan. This is Gulab. Nice to see you after
Hi.
2 years, right? Yeah.
Oh, welcome to California. I last
Yes.
saw you when you were moving to
I enjoyed your talk.
Alabama.
I just have 2 questions. One is, do you think this AA147 would work on a misfolded protein? Would it kick it out? I mean, if it sends it to the Golgi, it's still a problem. And then second question I had with this mutation for achromatopsia, the phenotype in the patient is just that, or because I'm assuming that beta cells which are highly secretive, this function of ATF6 is so essential. So, and why, I mean, I think Vladimir and others asked the question why these cones are going. Is it a developmental thing, you think? Have you studied that, that the cones don't develop well or something? Yeah, thank you.
Yeah, so with the first question, yeah, I think the activating the ATF6 especially is really great for just secretion of proteins, whether they're stuck in the ER or whether they're just normally and maturing in the ER. There's evidence, there's studies from a number of groups that it just enhances the secretion of efficiency of secretion and secretion of proteins out of the ER. So that could be really beneficial for disease proteins, like some of these ATF6 proteins that cause achromatopsia that get stuck in the ER, but it could also be beneficial for putting more receptor subunits on the surface or secretion, export of proteins outside of the ER. So there's quite a lot of papers, a number of papers in cell culture and experimental models that boosting activity of this pathway can just in general make the export and secretion of proteins, whether they're disease or function or wild type, more robust and efficient. With respect to the second question, why are cones so sensitive? Well, I think with the cones, it's a developmental defect. This is most evident in our retinal organoid model where the cone structures, the recoverant is fine. So we know that the cells are there, the nuclei are there at least. But really the defect is the formation of the structure on the organoid. It's not forming, and it's a really, really penetrant defect where the entire surface of the organoid is always remains smooth. With application of the drug that activates ATF6, and we're excited by that because you can just see that the surface becomes more bumpy, and that's corroborated with cone markers, outer segment markers, so on and so forth. What other cell types are sensitive to this? Well, our data, genetic data and our patient data supports that some of the other cell types that are sensitive to this at later ages are rod cells. So there are families with cone rod dystrophy where later on the rods are also deteriorating in function, and we think that ATF6 is also expressed in rods and could be playing a similar supportive role in their structure function. But also hair cells in the cochlea, they degenerate as well, leading to hearing loss in some of these achromatopsia sort of patients. And also in the ATF6 mice, there's the hair cells are degenerating. And those are also structurally and morphologically very similar to photoreceptors in that they're polarized cells with really, really specialized modified cilia structures with special proteins, and the absence of ATF6 leads to deterioration of that as well. In diabetes, the ATF6 mice, under normal conditions, they don't have diabetes, But then when you challenge it with conditions, drugs or conditions, or cross it with diabetic mice, it exacerbates those sorts of conditions. So it could play a role in the late downstream steps of progression of the disease. That's general true in almost all disease conditions where this protein's present in all cells, and its absence will sensitize to external forms of stress, external or genetic forms of stress. But in people, what's really interesting is that the cones are developmentally experience the first stress during development, and so those are the cells that are affected most immediately in people.
All right, Jonathan. So let me have a final question, and this will be exam from organic chemistry. So your compound 147 is aniline amide, right? So this is organic amide in a way. How stable it is? It should relatively quickly, even spontaneously hydrolyze, or could be.
So it actually within the endoplasmic reticulum, it forms a chemical reaction with protein disulfide isomerases to inhibit. So the way it kind of promotes ATF6 and secretion in general is that the secretion of ATF6 is dependent on full reduction of all the disulfide bonds, and that's dependent in part on protein disulfide isomerases within the ER, which there are a lot of them. This AA147 actually forms a chemical interaction with protein disulfide isomerases to inhibit their function. And in doing so, then that leads to increase in fully reduced ATF6 molecules that can then leave the ER. So that's how as Chris noted, that the structure of this is chemically reactive, and it's reacting with in the ER with protein disulfide isomerases, which in turn leads to ATF6 being fully reduced and being able to leave. Yeah.
Very well. So I have no option but to give you A+ in chemistry. Jonathan, thank you so much. It was a pleasure having you. We learned a lot today.
Okay, thank you. Bye-bye.